Welcome to the Ricci Flow Nutrition Podcast. My name is Cameron Borg. On this episode, I had the pleasure of speaking with Robert Fosbury. Robert Fosbury is an emeritus astronomer at the European Southern Observatory and an honorary professor at the Institute of Ophthalmology at University College London. For 26 years, Bob has worked at the European Space Agency as part of their collaboration with NASA on the Hubble Space Telescope. He joined this initiative in 1985, more than 5 years before the launch. During the latter part of this period, Bob served on NASA's Ad Hoc Science Working Group and the ESA's Study Science team as they developed the instrument concepts for the James Webb Space Telescope, the next generation of space observatory. Since then, Bob has become an integral part within the Institute of Ophthalmology at UCL with his unique perspectives on light and its interactions with the Earth's atmosphere. His interdisciplinary contributions to this team at UCL have led to new hypotheses developing, particularly surrounding the beneficial effects of specific wavelengths of red and near-infrared light and the detrimental effects of the narrow-spectrum blue light that is emitted from modern lighting fixtures and appliances. Bob and the team at UCL with Professor Glen Jeffrey are working hard to explore and share the damaging effects of blue spectrum light from artificial sources. Commonplace now, most artificial lighting peaks at wavelengths that have devastating effects on the energy production systems of the cell, the mitochondria, as well as having negative effects on circadian rhythm. Bob is extremely passionate and incredibly humble in his pursuits. I find his experience within this interdisciplinary team at UCL to be warming and very hopeful for the future of science. I had an amazing conversation with him that spanned over 2 hours, and I could have spoken to him for much longer. Bob is really easygoing and very willing to listen and learn. I feel very grateful to have had the chance to speak with him about his ideas because I strongly believe that they will be proven in the years to come. We got to cover a lot of ground in this episode with topics from astrophysics to red light therapy and particularly the dangers of isolated blue light. The key takeaway from this, from my perspective, is that the biggest public health crisis going on right now is artificial light. Hopefully, with work like his and Glen Jeffrey's, we will be able to turn this ship around and get to make light safe again. With all that being said, I hope you enjoy the episode. Thank you so much for taking some time to speak with me today, Bob. I've really been looking forward to this. I've been a little bit nervous to talk to you because this is well out of my wheelhouse, some of the things that we're going to be talking about. But you've got a lot of interesting ideas, and I'm really excited to hear your thoughts. Right or wrong, I think it's just great to, get some of these things out. So maybe you can talk a little bit about how you got involved in light, looking at light from your background in astrophysics.
Okay, I'll do that, Cameron. I'm outside my comfort zone as well. I mean, I'm not a biochemist. I know a bit about light, but I have got involved with the visual neuroscientists at UCL, and it's a slightly curious story, but I'll try and outline it briefly for you. I mean, as you know, I spent my career in astrophysics. I started at the Royal Greenwich Observatory in the late 60s on a summer course there. I eventually did a PhD at Sussex and the Royal Observatory and became a research fellow there for a short while, and then went on to Australia. Actually, I went to one of the first 2 fellowships to the Anglo-Australian Observatory. They just built this collaborative 4-meter telescope in Coonabarabran, in the Warren Bungles, and I was lucky enough with actually my office mate to get the first 2 fellowships, and so we spent time in Australia. Anyway, my career progressed from there through a number of stages: the European Southern Observatory, working at CERN, and then back at the Royal Observatory, working on the new telescopes on La Palma, and then going back to the European Southern Observatory, actually employed by the European Space Agency to work on the Hubble program, which I did for, I think, 26 years. So I was very familiar with the Hubble program. We collaborated very closely with NASA, of course. We were the European branch of the Hubble operation. And during that time, of course, this was the time of the gestation of what became the James Webb Space Telescope. And I was on one of the early NASA committees, the so-called Ad Hoc Science Working Group, that was tasked with selecting the kinds of instruments that should fly on the Webb telescope. And we did that in the late 1990s. And then I was involved in the beginning of the European, one of the two European instruments that flew on the Webb telescope, and this is the near-infrared spectrometer, which was actually built in Munich, which was very close, within a kilometer of where I lived at the time. And we were involved with the industrial contractors to begin to design and construct the instrument. Now, I didn't continue with that for very long because I had to go back to Hubble. But I did make one or two sort of very minor contributions to that, including thinking of a way to have an absolute wavelength calibration on the telescope, because the telescope has to be cooled to a very low temperature, 40 kelvins, and you can't use lamps for calibrating wavelengths because you can't dissipate the energy in the telescope. So I had to think of a way of doing that which didn't involve using any energy, and that was using a rare earth, a lanthanide in a filter, which provides dark absorption lines, which are very narrow, because the lanthanides have inner shell transitions in the lanthanide atom, and these inner shell transitions are protected from the surrounding Coulomb disturbances of crystals and so on, and so the lines remain relatively sharp. So it's almost like having an atomic clock inside an atom, and you can use that as an absolute wavelength calibration. And I suggested this in the early 2000s, and they actually adopted the idea, and it's actually sitting in the telescope. That was a minor contribution. That's a major contribution that one of my colleagues had it on the telescope, but maybe we'll come to that a bit later. Anyway, my career progressed until I had to retire from the European Space Agency. And I decided when I retired, I could see so many of my colleagues were basically just continuing what they were doing before, i.e. astrophysical research, without being paid for it, on a pension, so to speak. And I didn't really want to do this, not because I wasn't interested in astronomy, but I developed broader interests in light and color. In fact, I'd always been interested in light and color since I was a kid. And it was really the color of the sky that led me into astronomy rather than the astronomy itself. I was always fascinated with the sky and all of its colors. And this led to me becoming an amateur astronomer. So I, went through the usual path of building my telescope and so on. And I really wanted to be an astronomer. And I kind of made it into the field, which was fortunate. It was easier then than it is now, I have to say, in the 60s. But it was still not an easy task to get into a field where you really wanted to do research. But this interest in light and color remained with me throughout my period as an astronomer. And I spent a lot of my time, I have my own spectroscopes early on and now spectrometers, which I use to study a very wide range of things in geology, biology, and so on. I have a Flickr site which has lots and lots of spectroscopic observations in it of all kinds of things. And that was a kind of secondary interest of mine that I continued. So when I started to retire, I was invited by an astronomical colleague of mine who was professor of astronomy in Durham to apply for an interdisciplinary fellowship at the Institute of Advanced Study in Durham. And he said, well, we'd like you to come, but there's 5 days before the deadline and you have to get referees and references and so on. I said, look, there's no way I'm going to get referees and references in 5 days. You all have to write them for me. Anyway, I got onto this fellowship, which was 3 months in Durham, given the freedom of the university to go around and talk to people in all the different departments and faculties and so on. And that was a real eye opener for me to spread my wings and think about humanities, think about chemistry and geology and all of these things. And I kind of thought I should know more about the Earth and evolution and geology and biology and things like this. So I started taking an interest in reading up things, and I happened to be browsing the web, which I don't do very much, but I noticed there was a video produced by the BBSRC, the Biological Research Council at the time, and it was about reindeer in the Arctic. And this was somebody who had written a paper, had spent a lot of time in around Tromsø working with the Sami people in the Arctic. And they'd been studying reindeer vision. And he was from the Institute of Ophthalmology in UCL, next to the Moorfields Eye Hospital. And he'd spent a lot of time in the Arctic with local physicists and the Sami people studying the way reindeers see in the winter. And they discovered that reindeer, they induce a rather profound change in their eye during the Arctic winter. And because the light, obviously it's twilight, it's mostly twilight during the Arctic winter. The sun circles around the horizon, mostly, well, entirely below it in the 3 winter months around Tromsø. And so you get very long periods of twilight. Now, I knew that twilight was extremely blue, and I knew why, and I'd studied it myself many decades before. And I noticed that in the paper that these people wrote, they didn't mention why the reindeer had to change its vision during the winter. So I wrote to this guy and I said, I really like the paper, it's a very interesting topic, but you didn't mention why the reindeer had to do this. He said, if I may quote the email verbatim, he wrote back the next day and said, "Oh shit," he said, "I knew we should have an atmospheric physicist on the team." Now, I wasn't actually an atmospheric physicist, but I ended up joining the team. I mean, after some while, we exchanged emails and we exchanged Zooms. Well, it wasn't Zoom then. We Skyped one another. And eventually I started going to the lab and eventually started doing experiments on other reindeer eyes that he had. And, well, to cut a long story short, we sorted out exactly what happened in the eye to make the changes. And during that time, because I had difficulty getting into the building, because there's a lot of security in university buildings now, and so he said he'd get me an entrance card for this for the building, so I could get in without dragging him down from the 3rd floor every time. Anyway, the entrance card said honorary professor at UCL, so this gave me access to the library, which was fantastic because I didn't have access to a university library after I retired. So this was great, and so I did spend time there and worked on this, and he really wanted me to write a paper about it, and I was being very slow because I was trying to understand biology and so on. In the end, I did write a paper, and we submitted it this year, and in June, and well, no, actually we submitted it in March, and we had 2 referees who basically tore me to shreds because I didn't have the right language, I didn't introduce the context properly and so on. But these 2 referees were extraordinarily helpful, and they wrote very detailed comments about what I'd missed out and what I should have said and so on. So I took all these comments and I rewrote the paper, and it was resubmitted. And with great trepidation from me, because I'd never written a paper in biology before, and this was being submitted to the Proceedings of the Royal Society, which is not a minor journal. And I didn't really expect to get it through, but after I'd rewritten it, I got a note back from the editor saying you did a really good job of convincing the referees. So it was accepted. And when you write papers these days, you have to write a short when a paper is accepted in these journals, you have to write a short press release, 100 words press release. So I wrote a 100 words press release and sent it off and didn't think any more about it. But the week before publication, I began to get contacted by magazines and so on. So I got the Atlantic in the US and the Conversation in the UK saying, will you write or interview and get an article about the reindeer that comes out on the day of publication, which it did. And 2 articles came out on the day of publication, and in the journal they report the attention that this is given, and this came out as one of the top 5% of science stories around the world on that day. And I realized that writing papers on reindeer is much more popular than writing papers on distant galaxies. So I was in a new world, which was quite a shock at the time. But I mean, that was my way into collaborating in biology. So really, I think my contribution was to provide some kind of experience with the way light works, with a view to understanding how light interacts with biology.
Right.
Now, the real story, that was a slight distraction just to explain how I got into it. But the real story, when I started turning up at UCL to work on this stuff, they were working on the effects of red light on animal models. They were actually using fruit flies, Drosophila, at the time, and they were shining red light onto the fruit flies and looking at various metrics of their biology and behavior. And it was clear that this was producing a really rather profound effect on this animal model. I think they were also looking at other animals. They were looking to some extent at bees and also at mice, and later humans as well, not humans at that time. But this was very interesting. I thought this was extremely interesting. And I said, well, what wavelength do you use for this? And he said 670 nanometers. And I said, why are you using 670 nanometers? I mean, out of genuine interest. And he said, well, seems to work, and other people seem to be using it. And I said to him, I mean, my first words were, do you realize this is where chlorophyll does photosynthesis? And this just struck a chord with me at the time. And I thought, this is really interesting. And, I had learned that, the idea of photosynthesis and the origin of the chloroplast and the origin of the mitochondrion go way back in evolutionary history. I mean, we're talking about going back billions of years. So this was a connection to the very distant past. And I'd always been interested in the process of photosynthesis. I'd read books about photosynthesis. So my introduction to biology at this level was really through trying to understand photosynthesis, and in particular the history of the discovery of the various processes that contributed to it. And it was clear to me on reading about this that the physicists played a major role in this, partly with the use of isotopes to track molecular movements within the process to find out where the carbon came from and where the oxygen went and so on. And also when there was the problem of how the photon energy was transferred to the reaction center in the chloroplast. I was fascinated by this because I wondered whether it was to do with fluorescence, whether there was a fluorescent energy transfer, or how did the photon energy that was absorbed in the antenna pigments in the chloroplast, how did that actually get fed into the electron transport chain in the chloroplast to start the photosystem 2 in photosynthesis and get things moving? And as you probably remember, this was a problem that was actually solved in the early 2000s by doing high-speed laser tracking of the process, and realizing that there was I suppose it was one of the first realizations in biology that there was quantum mechanics was actually playing a role in biology, which shouldn't have been a surprise, but I think was a surprise to the biologists. And there was this very efficient transfer of an exciton in the antenna pigment into the reaction center in the chloroplast via the wave function of the exciton. And using the old language in quantum mechanics, the wave function collapsed at the reaction center and transferred the energy without this random walk through other antenna pigment molecules, which was thought to be going on at the time.
So my understanding of this was that it was assumed that quantum processes couldn't happen in biological systems because there was too many things to prevent any coherence from happening.
Exactly. The temperatures were too high. Yes.
Yeah, it was warm and wet, so it couldn't happen.
Yeah.
It had to be cool.
Yeah, right.
So when this was first proposed, it was basically laughed at that these quantum processes were happening. And I think it's like, is photosynthesis something like 36 quantized steps? Like it's a very, very careful process to get all the way down, which sounds crazy that it's quantum. But I mean, when you think it's interacting with photons of light, you're right. It's probably not that crazy now that you think about it.
Yes, it's a perceptual change. I mean, it's obvious that quantum mechanics is responsible for everything we do, of course. the way that the realization of that obvious fact takes quite a long while to sink into the broader scientific community. And I hope we'll come back to this question of the broader scientific community and how they interact, how people in different fields interact with one another, because I think that's terribly important.
Yeah.
But as far as the quantum mechanics of photosynthesis is concerned, that was a problem that was addressed and I think solved to a large extent by the physicists. And so that was an indication that one needed a very broad attack on these very complex problems in order to piece them all together. And photosynthesis is a wonderful example of that collaboration over many, many years, producing. I can't say that people understand photosynthesis perfectly. I don't think they do, but certainly it's understood at a very deep level now. Anyway, this germ of an idea of the connection between the wavelength used to study these processes in mitochondria and the process going on in plants in photosynthesis, it just stuck in my mind, and I'm not sure where it's going. My collaborator was a bit skeptical, and in fact, he used to get rather agitated when I talked about photosynthesis because he said, "My colleagues will never talk to me if they realize I'm working on plants." But anyway, that was something that really tweaked my interest in this, and then. Those experiments were going on. I wasn't directly involved in those, although I certainly discussed with his lab what I thought about the processes. So I was a kind of external sounding board for some of the experiments they were doing. But then they were talking about blue light. Blue light seemed to do the opposite. Blue light seemed to have negative effects on these animal models. And the question was, why was that happening? And again, there were some accidental collisions between ideas. These visual neuroscientists realized that blue cone photoreceptors seem to behave differently from the longer wavelength cones. And they had discovered that the blue cones contain fewer mitochondria than the medium and long wavelength cones. I mean, most of the cones in your eye are packed full of mitochondria because the photoreceptors actually use far more energy per cell than any other cell in your body. They use more energy than muscle cells. They use more energy per cell than brain cells even. Because to reload your photoreceptor after it's detected photons takes a lot of energy. And the puzzle was that the blue cones seem to use glycolysis. They seem to want to use the inefficient energy generation process of burning sugars to make some of their energy. and this didn't seem to make any sense. Why would they do this? Because it's inefficient, it's damaging to cells, and it didn't seem to be right. So we were thinking about why this might be the case, and we thought, well, why do these blue cones have so few mitochondria? And then I think I was doing a literature search, and I found a paper in the SPIE, the Society for Optical Engineering. I can't remember what the acronym is exactly, but it's a journal that's often used by engineers who build telescopes and instruments. So I was familiar with this journal, and the biologists were not at all familiar with this journal.
Mm-hmm.
And I found a paper which had actually measured the absorption spectrum of mitochondria, because they were interested in getting light energy deep into the body for various kinds of therapies. So they wanted to know what in the body absorbed what. And so they had a measurement of, they extracted mitochondria from the liver of a rabbit or something, I can't remember what it was, but and they shone light through it and produced an absorption spectrum. And this had a strong absorption at around 418 nanometers in the deep blue. So I sent this paper, copy of this paper, to the people in UCL, and it kind of blew them away. They didn't know this, and they said, well, if mitochondria absorb in the blue, and you have all the mitochondria in the light part, because the light has to go through that segment of the photoreceptor before it reaches the receptor itself. If it absorbs blue, maybe that's the reason that they don't have so many mitochondria, because they don't want to absorb too much blue light before it gets to the receptor itself. So we published a paper about that some years ago, and we actually also tried to extract mitochondria ourselves and look at the absorption spectrum. And we did this, and we got essentially exactly the same result, rather better result, but the same result as was discovered before. So it's clear that mitochondria absorb blue light, which, if you think about it, is not a surprise, because mitochondria have heme. They actually synthesize hemoglobin, so they have hemoglobin in their in themselves, and they also have cytochrome C as part of the electron transport chain, and they're porphyrins, and they have a Sorry band in the blue at around 420 nanometers. So it's no surprise that they absorb in the blue, but this was an example of a case where that blue absorption was a hindrance to the operation of the photoreceptor, so it had to modify its energy generation process. And so the group there got more interested in blue light, and as the worries about blue light illumination became more and more apparent, we became more and more interested in what the process was. Why was the blue light damaging, and what was it actually doing? And I think quite a lot of people are working on this now, and I think the consensus opinion is that the damage is done by reactive oxygens, and somehow the blue light stimulates this generation of reactive oxygens in the mitochondria.
Yeah. It was
was one.
It was very interesting, that paper that you sent me yesterday, the one from Scott Zimmerman and Russell Ryder. One of my favorite papers, actually. And he references a paper where they make the case that if you were outside in sunlight, most of the reactive oxygen species that you would make, or about half of them, wouldn't come from UV. They would actually come from visible light, which no one really gives any thought to at all whether visible light can have any health or beneficial or detrimental effects, because we can kind of see it, we're aware of it. There's not really any attention being called to it. But like you said, now there seems to be a consensus coming around that narrow spectrum blue light that is devoid of red light as well is very, very damaging. And like you said, the mitochondria basically just shut up shop as soon as they get shone with that blue light. I know that Glenn and one of his colleagues, who's now in France, have published work about how mitochondria just seem to say, no, I'm not going to do anything if you're going to shine with blue light that's not balanced by red. So, and just before I forget, you mentioned earlier that twilight is rich in blue light. Now, I was used to the idea that when the sun is on the horizon, it is more densely populated with the reds and the yellows and the pinks and the oranges. So how come twilight is more blue rich than I would have anticipated? If it's on the horizon, it would be more red in the red.
Well, when I say twilight, I mean twilight after the sun has set.
Yeah.
So we're not talking. Yes, the light from the sun, of course, is very red because all the blue light is actually Rayleigh and aerosol scattered out of the beam when the sun is on the horizon. You have to travel about 40 times the amount of atmosphere to reach you when the sun is on the horizon than you do when the sun is directly overhead. This is called the air mass. You have an air mass of 1 when the sun is overhead, and you have an air mass of 40 when the sun is on the horizon. So when the light is coming from the sun directly to your eye, you have to travel through this very large column of atmosphere. And the dominant process of getting rid of the blue light is Rayleigh scattering, and to some extent the scattering by aerosols, because Rayleigh scattering is much stronger in the blue than the red.
Right.
In fact, it's about 8 or 9 times stronger in the deep blue than it is in the far red. But aerosols also scatter predominantly blue light, although the ratio is not nearly as high. It's more a factor of 2 rather than a factor of 8 or 9.
Yeah.
So you very effectively distribute this blue light over the sky and remove it from the direct beam from the sun.
Right.
The thing that wasn't understood and is still not widely understood even by scientists is that the color of the twilight sky is blue, like the daytime sky. But the twilight sky blue is a totally different physical mechanism from the Rayleigh scattering of the daytime sky. When you look at a daytime sky, you're seeing Rayleigh scattered light, so it's a pure soft blue. I mean, gradually you get less and less red light as you go further, less and less light as you go further to the red. But twilight, this basically jumps to a different physical mechanism, which is one of absorption. Now, the strongest absorber in the visible spectrum in our atmosphere is ozone, which is in a layer between about 12 and 40 kilometers altitude. So we have very little ozone at ground level generally, without lightning flashes and so on. But we get a lot of ozone high in the atmosphere, and we all know about the ozone hole and so on. It's high atmospheric physics that produces the ozone. And ozone is blue. If you look at liquid ozone, it's a gorgeous ultramarine blue, very rich blue. And that's because we know about the absorption of ozone in the ultraviolet part of the spectrum because it allows us to survive. It absorbs all of the damaging, or most of the damaging ultraviolet light, and allows life on the surface to survive. But there's another absorption centered in the orange called the Chapuis band, after after Chapuis in I think the early part of the last century. I can't remember exactly when. And that band is generally very weak when the sun is high in the sky. Astronomers know all about it because they have to measure it to look at atmospheric extinction of starlight. But most people are not aware of this absorption band in the visible spectrum in the orange. And it's a very, very broad band. It's a very unusual looking band. It's complex in shape and it's very, very broad. It's centered quite near the sodium D lines in the orange, and it spreads into the red, and it spreads right down through the yellow into the green, and is very weak by the time it gets to the blue. So you get this big, very, very big absorption band, which as the sun sets, as we said, the path length through the atmosphere increases.
Mm-hmm.
This band becomes increasingly strong as the sun gets lower down in the sky. And it begins to become noticeable in the color of the sky around about the time of sunset, even when the sun is still above the horizon. If you look at the sky is taking on a color which is strongly influenced by the ozone. In fact, it's remarkable that if you model the sky, the color of the sky, with just these scattering processes, ignoring the absorption by ozone, the color of the clear twilight sky at sunset would be straw yellow, slightly grayish straw yellow.
Wow.
But if you look at the zenith at sunset, it's an incredibly deep metallic blue-purple, and this is entirely due to ozone.
Mm-hmm.
And this was discovered in the late 1940s and early 1950s by the American atmospheric scientists who were firing rockets donated to them by the Germans after the war into the high atmosphere to study the color of the sky and the strength of radio scattering and so on. And there was a guy called Hulbert who was flying these rockets, and he'd calculated the brightness and color of the twilight zenith sky using radio scattering models of the atmosphere. And he found that the measured color and brightness was very different from what he computed. And this was a real puzzle around the late 40s, around the time I was born, actually. And he published a paper in 1953 about the color of the twilight sky, showing that it was due to ozone gas absorption and this Chapuis band. Beautiful paper, described very clearly with lots of models and so on, completely solved the problem. But of course, nobody knew about this paper. I didn't know about this paper.
Right.
I studied the color of the sky myself, entirely independently, without any knowledge of this paper in the early 90s. And I came to the same conclusion. I realized by looking in my astronomical reference books that there was this absorption band in the orange called the ozone Chapuis band. And I modeled it myself. And I made this discovery entirely independently in the early 90s. And it was many years later. I mean, I didn't think terribly much about it. I thought, okay, well, that's the answer. But I didn't know the history at all. And I was sitting down with colleagues in the Space Telescope Institute in Baltimore. I spent a lot of time there. And the person I was talking to at the time had studied atmospheric physics under somebody called Goody. He was the father of atmospheric physics. I've got his book here.
Wow.
And this guy did his PhD under this guy, and he remembered something about ozone. So we looked up in the book, and he found this reference to ozone. So it was a couple of decades later that I discovered that my discovery in the early 90s had been done by Hilbert in the 1950s. So I became very fond of this problem. I mean, I was attached to this question about ozone. It also has many implications in art and poetry and so on, because the artists call the hours of twilight, or the hour of twilight, the hour blue, l'heure bleue. It's a time where artists are aware of the fact that there's special atmosphere of the light, special quality of the light at these times, and they realize what it was. But your eyes don't really see this, because it's the time when you're transferring to your rod vision, which is, of course, colorblind, And also, your eye is very good at adapting to changes in color. As
Yeah.
you know, you adapt to a color, so you don't see it. You see it as gray rather than blue. But if you take photographs of twilight with a daylight color balance set on a white balance set on your camera, you'll find that half an hour after sunset, the sky and the whole environment is incredibly blue. You'll think you made a mistake with your camera. In the old days, you made a mistake developing the film because it's so blue.
I guess it's somewhat confusing, or at least you could say I don't understand it, because um, you would think that the wavelength that has the biggest impact on something as simple as the pineal excretion of melatonin at night wouldn't be present at night. So I'm wondering whether the intensity of that light is very high, or it's just that its relative abundance to the other wavelengths is so high.
Well, you raised two very interesting points. I think twilight is quite faint, of course. The intensity is down by factors of tens of thousands from daylight. So the intensity is very low. But it is also true that the balance of lighting is very different. And coming back to the reindeer for the moment and the adaptation of the reindeer adapts to see this the residual light after the ozone has absorbed the rest. And if you look at that spectrum, you find that it peaks very strongly in the blue at about 450 nanometers. And this is exactly the wavelength of the reflection from the reflector behind the retina of the reindeer, which gives the photons a second chance of going through the retina. That reflector, called the tapetum lucidum, the shining carpet behind the eye. We don't have them in humans, but they have them in many animals, including all the bovines and the ungulates. And
if I remember correctly, there are pictures of this structure in the paper, and they are absolutely stunning.
They're stunning, yes. The twilight blue is really quite something.
Yeah.
And this is, it's fun to observe. And also the other phenomenon that you can see at that time is if you have a clear sky. Let me just emphasize that for the twilight blue, the blue hour, it doesn't matter whether the sky is clear or not. It's always blue because at twilight, because this ozone acts like a color filter arching over the sky, and it just changes all the incoming light to blue. And so, unlike the daytime sky, when you only see the blue when the sky is clear, at twilight, the sky is always blue. So the reindeer have to live 8 or 10 hours a day under this rather low but significant blue illumination. And this was presumably the cause of their adaptation. They changed their reflector from a sort of golden color in the summer to a blue color in the winter. We worked out exactly how that change happened. I won't go into that now, but we understand exactly what the physical change was in the structure of this mirror behind the retina to do this. But yes, and there's an interesting point you make about the intensity and the color of the twilight. Talking to my circadian biologists and physiobiologists, they think that change in color at twilight is perhaps the strongest, clearest, most reliable signal for this setting the circadian clock. The brightness of the sky is very unreliable because it's so dependent on atmospheric conditions and so on and so forth. But the color change always happens, and that's a very, very powerful, reliable signal of the time of day to set the circadian clock.
Yeah, I believe there are actually cells, if I remember correctly, the intrinsically photosensitive retinal ganglion cells that are the ones
that's right. Yes,
that
those are the ones that are
Yeah, they assess the color temperature, so that that you see a lot of people now in the health and wellness space saying how important it is to get at least 10 to 15 minutes of early AM light,
Mm,
and then light on the horizon as the sun is setting, as they are the
mm.
most important times of the day to set the clock. And I think that's an incredibly powerful tool to use. And um, yeah, like you said, with all of these circadian guys, they know that that's the prime time. I mean, also getting bright light during the day, the brightness is also a factor. But I
Yes.
think these color temperatures that you see in the early morning and late afternoon are very important signals for the eyes and the brain.
Yeah, well, I mean, let's try and unpick that a bit. There's a
Hmm.
lot going on here, and I think we have to be careful about the different things that are going on. The circadian business is to do with controlling the functions of your body at different times of day and so on, and there's a huge amount of research that goes on in that area. And the person I speak to is someone called Manuel Spitzen, who was at Oxford until recently, is now in Tübingen and Munich working on this. He's the guy who organised the summer school that I went to this summer, and he was doing very interesting experiments on this circadian switch to do with the blue twilight, and he was looking at the reaction of eyes to absorption in the blue at slightly longer wavelengths, around 490, 500 nanometers, that interacts in the melatonin business, and the effect of that on the eye, and also the effect of deeper blue light on the blue cones. And so the melatonin process goes on in these ganglial cells, and the deep blue process goes on in the cones, in the blue cones. And he was looking at the reaction to the cone signal and the ganglial cell signal, and they work in opposition to one another. Now, I won't try and explain that to you in detail because he's the expert, and I'm not sure that I could explain it carefully. But they had those two signals had opposite effects on your pupil dilation,
Hmm.
and I think the story went that this opposition between these two signals at different blue wavelengths produce this very strong trigger for the setting of the circadian clock. So there's a lot going on with melatonin and circadian processes to do with the quality of the light in the blue, which is all very interesting and needs to be communicated more widely between them and other biologists, I'm sure. But I'd like to emphasize that that set of processes is somehow distinct from the pure interactions of photons with biochemistry, with individual molecules that we're talking about with the red light and the blue light and the reactive oxygens. Now, the way of making the reactive oxygens from the blue light that
Yep.
you mentioned, this fact that it's blue visible light, not far ultraviolet light that has this effect. When you excite a porphyrin with blue light, you change you induce an electronic transition within the porphyrin to a higher excited state. And in fact, with blue light, I think you excite to the second electronic level, and that excitation can result in a radiationless transfer to another higher level, a bit lower down, which is metastable, and it hangs around for a while. And while it's hanging around in that excited state, this porphyrin can interact with an oxygen molecule and can change the excited state of an oxygen molecule from a triplet state to a singlet state. So this is a quantum mechanical process that happens when oxygen and porphyrin molecules come close together. And this can create singlet oxygens, which are known to be very highly reactive. And these are the reactive oxygens that in the early days of photodynamic therapy, when people were killing cancer tumors cells with with light.
Yeah.
This was the reactive oxygen, the singlet oxygen that they produced using porphyrins and blue light.
Yeah, right.
Now, that was the early experiments going on in this field. And the problem was that they couldn't penetrate the blue light. The blue light wouldn't penetrate into the tumors. So it was very hard to get the reaction occurring within the tumor cells so you could kill them. It tells the cells, it doesn't burn the cells, it tells them to commit suicide. So what we're doing with the blue light, we're generating through the oxygen, we're generating reactive oxygens that are potentially damaging. I mean, the reactive oxygens are used for other things. They're used for signaling between cells and so on. So they're a central part of the biological process. If you get too many of them, they do too much damage. And so that's a problem. So we do know there's a pathway in the blue, which is a sensitive function of wavelength. It's just in this range in the deep blue, where you can excite the porphyrin and you can produce a reactive oxygen. Now, there are some subtleties here, which I'll try and explain, but in mitochondria, we know that mitochondria synthesize heme. And to synthesize heme, you need to have a precursor molecule, which is something called protoporphyrin 9. So this is a porphyrin that doesn't have a metal in the middle. Now, to make heme, you have to add iron into there's an enzyme that does that. That's how you go off and synthesize heme. That same protoporphyrin molecule is the immediate precursor to chlorophyll. So in chlorophyll, it's porphyrin like protoporphyrin like heme, but instead of having an iron, it has a magnesium in the center. So this protoporphyrin molecule is the precursor to both heme and chlorophyll, the colors of life. There's a wonderful book by Milgram about porphyrins.
So presumably that protoporphyrin 9 is highly conserved from many millions of years ago.
Absolutely, yes, absolutely. And it's a photoactive molecule, and it was realized by the photodynamic therapists that they needed photoactive molecules because this photoactive molecule, when it's excited, produces reactive oxygen. So it's this porphyrin that produces the reactive oxygen. It's not the hemoglobin or the chlorophyll itself so much. It's this precursor porphyrin that is such a generator of the reactive oxygens. And it explains in this book exactly how it happens. It's rather elegant, but it's deep quantum mechanics in a way. So we know that mitochondria at some time in their life cycle will contain protoporphyrin because they use it to generate heat. We don't know exactly what time of day that happens. Those experiments ought to be done by somebody, possibly by us, but it's we're not terribly well equipped to do that. But we know that protoporphyrin is present, and we assume that while that protoporphyrin is present within the mitochondrion, it's very sensitive to blue light. And so if we shine blue light on a mitochondrion while it's going through this process, it will cause damage.
Do you have an inkling when the mitochondria will have the highest concentrations of this protoporphyrin?
Well, I think we rather naturally assume that it's in nighttime because we know the mitochondria kind of restructure themselves and rebuild themselves all the time. But they probably do this at night, and when they're not expecting any blue light, it's a safe time for them to do this. So we assume that it happens at night. So now we start shining blue light on everything at night.
Yeah,
we're doing a lot of damage, and this is probably the dominant mechanism for the blue light problem. It's the fact that we're shining. It's not the blue light itself. If you do this at daytime, it's balanced by other processes, especially in the red. But if you shine blue light on mitochondria at night, you're not doing it any favors. And this is probably the origin of the problem.
I was going to say, it sounds like from a very, very from a 1 foot view that blue light is bad, full stop. But clearly, we get lots of blue light during the day from the sun, but it's the dose that makes the poison, and presumably that red light, particularly in the near infrared, is going to be balancing any singular oxygen that is made in that way. I think I've read, it might be in that Zimmerman paper, where the near infrared sort of acts as like an antioxidant. It has an antioxidant mechanism sort of built in. So yeah, that balance seems to be very, very important.
I think that's right, and I think that's a picture which is beginning to clarify. I'm not sure that anyone understands all the mechanisms, all the actual biochemical, physical mechanisms that are contributing to this process. But yeah, if we have a sort of mid conversation recap and summary,
Yep.
I think the point is that we've evolved over including pre mammals. Life has evolved for the last 2.5 billion years, ever since oxygen entered the atmosphere. We've evolved to live with oxygen in the atmosphere. And, oxygen up until that time had been extremely toxic to life forms. So life forms had to get used to having oxygen in the atmosphere. And of course, the oxygen was produced as a waste product from photosynthesis. And so the photosynthetic organisms, the plants and the cyanobacteria as their precursor and so on, lived with oxygen, and they lived with the production of reactive oxygens, which was associated with the photosynthetic process. So we've lived with this, and we've lived with sunlight and thermal light sources over these billions and millions of years. And we've adapted to the dangers, and the dangers are associated with oxygen. And so, we entered the last century with a valid balance of light and oxygen in life and in our own lives. And this balance seems to be associated with the damaging effects of the blue light on one hand, which we've just discussed, and the mediating effects of the red light, which we haven't yet discussed in any detail. But the fact that if you look at the spectrum of the sun and the other hot sources, the sources that produce thermal radiation, black body spectra, there's a very good balance between blue and red. In fact, daylight and sunlight have almost equal energies per unit wavelength in 420 nanometers and 670 nanometers.
Yeah.
So they're very well balanced. And then we started saying, well, it's very wasteful to generate all our light using thermal sources, because most of the energy comes out in the infrared. So we better think of something better. And so the lighting engineers, first of all, thought about fluorescent tubes, which work in a very different way from a glowing filament. And so they produced fluorescent tubes, which I think are generally acknowledged to be horrible to work under. They don't have they have very little red light, and they don't have any red light near 670 nanometers.
Yeah.
And so, they have quite strong blue light. So they're kind of uncomfortable. Nobody was really sure why, but they were kind of uncomfortable to live under. People felt terrible when they walked out of a whole day working in a factory with
They flicker like mad as well.
And they flicker as well. All kinds of things are wrong with those. And then they thought, oh, well, we can do better than fluorescent tubes. We can make LEDs, light-emitting diodes. And then the Japanese won this Nobel Prize in 2014 for the wonderful blue LED, which was going to revolutionize lighting throughout the world, which unfortunately it has. And this was a mistake. I mean, it's not a mistake made by the physicists who won the Nobel Prize, because it's a wonderful invention. Physically, it's a wonderful invention, has many uses.
Yeah.
But to use it for lighting is a disaster. And it produces extremely strong blue light, which is its only light product. All the other colors are produced by phosphors inside the structure of the light, of the bulb, which produce the other colors. But because lighting engineers quite reasonably thought that, well, we don't want to waste any energy, so we'll only produce visible light in our devices. And these LEDs are extremely good at producing only visible light. They don't produce any ultraviolet, and they don't produce any infrared, and they're extremely efficient at producing white light, which costs very little to run, which means that councils buy them and they make them far too bright. We don't need, we can reduce them by a factor of 10 without, damaging their effects. So everybody produces much too much white light, which contains this enormous blue peak at a wavelength which doesn't exactly coincide with the protoporphyrin absorption, but it's close enough, and both absorption and emission bands are so wide that they very significantly overlap. And I can't emphasize too strongly that this is a mistake, and it has to be changed. It has to be changed. We can't continue using these very white lamps. We talked a bit about color temperature early on. The color temperature of the light outside my window at the moment is 4,000 kelvins, which means that blue peak is extremely strong. If you buy a light bulb in the store and take it home, don't buy a 4,000 Kelvin light. Buy a 3,000 or a 2,700 Kelvin lamp, which has a much weaker blue peak and much stronger emission of the other colors in the visible spectrum. But none of these LEDs contain significant infrared light. And I think this is a fundamental problem. And I think this is what we have to change. And it's not a problem for the lighting manufacturers. They can easily make lamps which include significant infrared emission. By the measure of efficiency of producing producing only visible light, which you can see with your eyes, they'll be less efficient, but they'll be considerably more efficient at making you feel better and not killing you. So this is I'm serious.
No, I know.
This is a slow death. The reactive oxygens that are produced by blue light are the same as the reactive oxygens produced by Chernobyl radiation. It's the same process that kills you. Okay.
I genuinely believe that the biggest public health issue we're facing right now is our lighting. I think
it is. It's
without a doubt.
Insects.
And like you said before, it not only affects the circadian system, but it's also causing direct damage. So you've got these two pathways in which it's absolutely destroying us.
Yes.
And I know I asked Glen, what are you looking to do? Like, what are you moving towards? And he basically said, what I want to have on my tombstone is that we changed public lighting. We got them to fix the mistake. And that was a very, very powerful message to me that, it really is that big of a problem. It's not minor, and
the insects that we use on these experiments are fruit flies. Okay. Maybe we shouldn't do animal experiments, but then we use fruit flies. Okay. If you shine blue light on fruit flies, first of all, they don't have any energy left. They can't climb up the cells, the tubes that we put them in. And if you shine too much blue light on them, they die. But anyway, it reduces their lifetimes. If you shine red light on them, they have loads of energy and they live longer. The insects, we're killing the insects around the planet by leaving these bright white lights on all night. And I can't say whether this is the major cause of the insect loss around the world. It's probably associated with insecticides as well. But the light certainly isn't helping, and it could be a very significant factor in killing the insects around the world. And all we need to do is to repair the balance between the spectral colors of the light that's emitted by the devices we use at night. Now, this is not, I hate to use this phrase because it's so silly, but it's not rocket science. It's easy to do. The manufacturers can do it at the drop of a hat. They may produce devices which are slightly less efficient, and they may cost a little bit more, but they can do it and they'll save the planet. This is called, but I heard the expression once, this is the new asbestos. The white LEDs are the new asbestos. We're going to have to root them out of buildings and change everything before they kill us.
Yeah.
So we feel very strongly about this, but that's in a way the fundamental driver. But I'm particularly interested in the processes that contribute to this problem. Now, I'm not equipped as a biologist or a biochemist, but I do think very broadly and deeply about this. And I think the evolutionary connections are very important. It's not only the evolutionary connection with the chloroplasts and the mitochondria, which go back to the first eukaryotic cell. It's other chemicals, porphyrins we've mentioned. The melatonin is a very ancient molecule. It way predated the first eukaryotic cell. So this molecule goes way back in evolutionary history. And as you hinted before, it's a very powerful antioxidant itself, and it seems to stimulate the production of other antioxidants as well. And I remember watching another one of these YouTube videos by Professor Schult, S-E-H, can't remember how it's spelled, who has been pushing this idea of the effects of light on health. And he likened this to an engine. If you think of the mitochondrion as an engine producing energy for your cells, when it works hard, you have to cool it because otherwise it slows down and stops. And the analogy is that, as it's working, it produces lots of these reactive oxygens as a byproduct of actually producing the ATP, the chemical energy that is needed by all the cells in the body. And the coolant is the antioxidant, which mops up these reactive oxygens. Now, this comes to the area which I think is very interesting at the moment, and I have to admit that I don't understand exactly what's happening. I'm not sure that anyone understands exactly what's happening, but in your favorite paper that you pointed out, I sent it to you yesterday. I'm glad you already had it. But it seems that the infrared light somehow stimulates the production of melatonin within the mitochondria. And, there are various references in these papers which point to the fact that this happens, but I've not been able to find or figure out the actual physical biochemical process that links the infrared radiation with the stimulation of melatonin production. I don't know if you have any ideas about this, but
I found today that last month Ryder and Zimmerman published a paper that I think directly looks at that question.
Right. Okay.
And they talk about how sunlight and exercise both increase mitochondrial production of melatonin because of the excess reactive oxygen species that generates. And if you put rats in a very stressful situation during their pineal melatonin peak, all of that pineal melatonin disappears from the blood very quickly because it's gone straight into the cells to prevent the damage from the reactive oxygen species. So I think they're pushing now not only this idea that blue light should be avoided at night, but that bright sunlight and exercise should be you should expose yourself to those during the day to help upregulate your melatonin production, your mitochondrial melatonin pool. And I'm not sure how that translates to greater peaks of melatonin at night when you're asleep. Apparently it does, though. I'm not sure how trustworthy or how many of those studies have been repeated, but it seems as though exposure to these red light panels does increase nocturnal melatonin secretion from the pineal gland. I'm not sure how that really happens. Scott Zimmerman seems to be pretty confident that that's how it works. When I asked Russell about this, Russell seemed slightly more apprehensive to say that's exactly what's happening. Classic, scientists being very reluctant to say 100%. It does appear that that's happening, but he was a little bit more reluctant, saying, yeah, I think we still need to do a few more studies and figure this out a little bit more. But I mean, I've probably not come across anyone who hasn't benefited sleep-wise from exposing themselves to 660, 670 and greater nanometer light, particularly at night. So clearly there's something going on. I'm not sure if we understand what it is, but yeah, they published a new paper like a month ago that I think
I'll look at that. I haven't seen
this.
that, but I'll look at that.
Yeah.
So. No, I mean, it is fascinating, but I still don't. There's no sign of understanding this mechanism. What's the light absorbed by, and how does that link to the biochemical changes? I think that's what we want to understand. And there's a way of describing this, which is well understood in photosynthesis. You want what's called an action spectrum. You want an experiment that allows you to shine light of different wavelengths, different colors on a biological system, and a metric to record what happens within the biological system. And a lot of work's gone into this in photosynthesis, and you get special photosynthetic lights which shine the most appropriate wavelengths onto the plant to either make it grow or make it flower or make it fruit or whatever. So we need an action spectrum for light on animals as well as plants of all kinds, anything from yeasts to humans. And, there have been attempts to do this. There was a Russian lady called Karu, I think, who worked extensively on this idea of a biological action spectrum.
I think she discovered that cytochrome C oxidase had the absorption spectrum in the
yes, that's right. Whether that's I think there are probably a number of different processes that contribute to this overall effect of the red light. And I think, in my view, the way to think about the red light is not so much that it results in one particular biophysical, biochemical process, but the fact that red light can penetrate living tissue, plants and animals. So we have between about 600 nanometers and 900 nanometers where water kind of takes over in the absorption. We have this fairly broad range of wavelengths where bodies and plants are relatively transparent. And in the paper you were referring to, there's a lot of discussion now of the way that this red light can penetrate into the human body, the way it can penetrate into the brain, and the way it penetrates deeply and reaches a large fraction of the number of the cells in your body. So I think that wavelength range is absolutely critical for the way the light interactions occur. The eye is unique in that we can get blue light into the eye. So the blue light process is naturally studied in the eye where it reaches the mitochondria. It will reach mitochondria in the skin. In fact, I think Glenn probably, in his interview, probably mentioned to you this experiment that was done with sunbeds converted to blue rather than ultraviolet sunbeds. It's a German experiment that was done a number of years ago. And they looked at they put young active people in sunbeds and shone blue light on them for a while and looked at their blood circulation and blood pressure and so on. And there was a dramatic reduction in blood pressure in these people in these blue sunbeds.
A reduction
in blood pressure.
Right.
And the researchers thought, this is great. We found a way of reducing blood pressure. Now, we think what happened in this experiment was they were, by producing reactive oxygens, they were inducing hypoxia in these people.
Yep.
And blood vessels were dilating to increase the blood flow to get more oxygen into the cells.
Interesting.
So they actually not killing these people, but they were damaging these people with the blue light. And the reaction was to lower the blood pressure and try and get more blood, into the cells to respire properly.
Well, this is something that I heard you talk about in an interview that I saw. There's a video of you on YouTube, where you talk about this idea that excessive blue light has a relationship with this hypoxia, like not allowing oxygen to be available. And I work as a pulmonary technician, so I work with a lot of people who have, sometimes chronic hypoxia, but at least transient when we do testing. And I wonder, because we're under 4,000 Kelvin blue LEDs, I wonder if being in a room like that is not very friendly for people who have respiratory issues.
I bet you're right. Yes.
Right. Okay. So it is having that much of a difference, as an impact rather, on the way that our cells are being oxygenated. Also,
I think switching to very fit people, for sports people who are working, who are playing sports at night under floodlight. These floodlights tend to be very high temperature LEDs. I mean, they've just put some 4,500 Kelvin LEDs in the Bath University sports pitches, and these are probably damaging for the sports people, because this is certainly not helping them respire at night. So I think there need to be experiments with much lower color temperature floodlighting for sports activities at night. And there need to be experiments. I know people are thinking about doing experiments like this. It's not very easy to do, because if you try and test people after they've played a game of football or something late at night, they're not going to be very willing to cooperate, I don't think. But I think this must play a role. It's very likely will play a role in performance. So, and it seems to me that, as you suggest, this production of reactive oxygens somehow glues these oxygens down and stops them getting to the places they have to be to take the electrons from the electron transport chain and make ATP. I think you're blocking the oxygen flow to the right places. Exactly where and how you're doing that, I'm not really capable of figuring out. But I think there is a process that's stopping the oxygen getting to the right places. And I think conversely, I get the impression that the red light may be actually doing the opposite. It may be freeing up the electron transport chain in some way, which is allowing the ATP production to occur more efficiently, more quickly. And some of the animal experiments that have been done have shown that, in fact, Glen mentioned recently he's done some experiments this way, or he's seen the results of experiments that have shown this, that these animals lose weight when you shine red light on them, because they're respiring much more quickly. They're being much more energetic. They're being much more efficient in using their food to respire and produce ATP, and they lose weight. And that's not damaging for them. In fact, it's good for them, because they don't become obese. So this is another potential application of red light, is to increase people's ability to respire properly, and affect levels of obesity. So there's another potential application here, I think. In fact, if we have time, I'd like to come back to a couple of experiments which were done in the US at Columbia over the last decade by somebody called Elias Washington. I came across these. Glenn showed me these papers some while ago, and I got very excited by them, because they were experiments on nematode worms, where they were shining red light on nematodes and monitoring their ATP production. And and very nice series of experiments. And their conclusion was that if they fed them on chlorophyll-rich diets, which is probably the norm for nematodes in the soil, they were eating plant, decaying plant matter. If they were on chlorophyll-rich diets, the application of 670 nanometer radiation to them for a while would increase their ATP production. And they did the sums, and it sounded very convincing. But their hypothesis was that the chlorophyll was coming through the gut wall and was, getting into the cells and into the mitochondria. And I don't think anyone believed this. And I think these papers were not referenced; they were ignored. Right. And the reason was, I understand that the biologists thought it was impractical to get chlorophyll breakdown products into the mitochondria. Now, I was very struck by these papers because they really showed all the results that we observed ourselves in flies and so on, and went into a great deal of detail about how the process worked. They did fluorescence spectroscopy of the mitochondria, and there was an emission peak in the fluorescence spectrum, which looked to be associated with a chlorophyll breakdown product. So they had a lot of evidence that what they were saying was sensible and correct. And I looked at this for a long time, and I looked at the sizes of the molecules and whether they could possibly get through the porphyrins into the insides of mitochondria. But it's very hard for me to judge what the possibilities were. And then I was talking to one of the photodynamic therapists at UCL, who has done a lot of work on producing photoactive molecules that work with red light, because this is what they need to penetrate cancer tumors and get the light in to do the cell suicide business. And he pointed me to a paper which I looked at, and this showed a fluorescence peak at the same place as the one that Washington had recognized in the nematodes. But this was produced by what's called a photoproduct of protoporphyrin. If you shine visible light on protoporphyrin, you convert some of it to something called photoprotoporphyrin, I call it PPP for short, which is a chlorine and absorbs at 670s and has a fluorescence peak, which was, as far as I could tell, I could measure this in my, I could measure this in porphyrins. And I compared it to his fluorescence peak, and it was identical essentially. And the thought struck me that if you had protoporphyrin in the mitochondria, and the mitochondria had been subject to visible light, it could have generated this PPP within the mitochondrion itself. So you didn't have to import it, it was actually generated inside. And this could act as an antenna pigment, which would absorb 670 radiation and possibly feed it into the electron transport chain. And this is where I get lost because I can't do experiments, but it's very analogous to photosystem 2 in photosynthesis. Mm-hmm. You have an antenna pigment in the chloroplasts. You absorb the 670, 680 nanometer light. It produces an exciton, which gets transferred to the reaction center in the mitochondria, in the chloroplast, and produces pushes the electrons up the electron transport chain. Why couldn't this happen in mitochondria? Now, mitochondria are obviously closely related to chloroplasts. Whether they have anything which could act as a reaction center, I don't know. But this could be a process that could work in a way which is analogous to photosynthesis. But instead of going on to use this potential across the membrane barrier in the mitochondria, instead of using that to go on to photosystem 2 and make sugars and so on, you could actually just use it to make ATP. So maybe that, I'm not saying this is right, but maybe there's a core of an idea there that you can actually generate an absorber in the mitochondrion, which may be able to transfer photon energy in some way into the electron transport chain.
So if
So
I'm hearing you correctly, you're saying something along the lines of we can generate energy directly from light, in a somewhat similar way that that plant cells generate energy from light.
Maybe, but I'd qualify that by saying it's not the hypothesis that you can't. Perhaps we know you can't generate enough energy from light.
Yeah.
In bodies, because we use energy far too quickly. I mean, it's not feasible to hypothesize that you're generating a significant fraction of energy. But maybe what you're doing is freeing up the electron transport chain. It's more like a lubricant on the electron transport chain to allow it to produce, to allow it to respire more efficiently. So I think what I'm saying is it's in a way, it's a catalytic process. It's not a fundamental source of energy, but it's a way of making the respiratory production of energy within the electron transport chain work more efficiently. You see what I mean?
Yeah, yeah. So it's not, you're not going to.
It's not a significant amount of energy.
Yeah. So, just backtracking to these chlorophyll metabolites. I know there wasn't very much follow-up from that initial paper that sort of made the case that you could generate energy from these chlorophyll metabolites interacting with light. I've asked one of Glen's colleagues about this, and he was unsure. He said it would be great if that was true. I personally, I take a little bit of chlorella before I do any sort of red light therapy if I'm going to the sauna or anything, just in case that is true. But I don't know. It seems like it could be possible that there are molecules from the things that we eat that do make it into the cell that interact with light. From my understanding, there are things in like parsley and coriander that these celarins that interact with light, and they can make you a little bit more UV sensitive. But it seems quite plausible to me.
Well, I'm not proposing this terribly seriously, but I think it opens up an idea that we should look at processes like this. And again, it's a broader view. There may be a mechanism which somehow accelerates the production of melatonin in the mitochondria with infrared light, maybe a broad spectrum infrared light, I don't know. But that seems to be established, or beginning to be established anyway, although we don't know exactly how it happens. But that would provide a process, a mechanism by which you produced antioxidants which countered the reactive oxygens that were being produced by the energy generation process. So that's one process that could operate with this red light that penetrates the body. There may be other more direct processes like this, which we don't yet know about or haven't yet established, which work in a much more analogous way with photosynthesis. I think that's what I'm proposing. And all I'm saying is that I've seen one mechanism where this could conceivably work, and I've done experiments with this on I don't have a lab here. I have a study which has a square meter or so of space to work on. But I did some experiments where I took hen's egg shells, which contain lots and lots of protoporphyrin, and shone light on them for half an hour or an hour, and then looked at their fluorescence spectrum. And they have if you take a box of brown eggs into a dark room and shine a UV light on it, they glow brilliant red. Have you done that experiment ever?
I haven't, but I have seen it.
You must do it. It is spectacular.
Right.
If you take a box of eggs into a dark room and shine a long wavelength infrared light on it, they glow a brilliant red. And this is protoporphyrin fluorescing.
Yeah.
But if I excited those eggs by shining light on them before I did the fluorescence experiment, a new fluorescence line appeared in the spectrum, which coincided with Washington's fluorescence line and the fluorescence line that comes from the photodynamic therapist paper. So I had generated protoporphyrin,
Yeah.
PPP, in the eggshell by shining light on it. In fact, I could write onto the eggshell. I took a multiple red laser dot and shone it onto the eggshell, and then subsequently photographed it under UV light, and you could see the spots. So I could write onto the eggshell. I could have written a message onto the eggshell with light.
That's incredible.
So in the eggshell, I could produce PPP from protoporphyrin.
Mm-hmm.
And there's no reason why that couldn't happen inside a mitochondrion. You have the protoporphyrin. If you have the light, you can do it. So, and we know this absorbs in the, we know this absorbs like chlorophyll. So, that's one mechanism. Maybe it's not right, probably isn't right, but I just thinking in that way, I think there may be a number of mechanisms by which light interacts with biomolecules in particular ways, and we don't yet know what all of those are, but they may be there and they should be looked for. And I think one way of trying to find them is to try and get an action spectrum of light interacting with cells which contain mitochondria at different wavelengths. And that's what we're doing at the moment. But we're building an experiment in City University at the moment. I found an engineer who was willing to collaborate with us. He's given me some lamps and gratings and so on. So we've constructed a prototype device where we can put samples of yeast or bacteria under at different wavelengths along a projected spectrum at about environmental light levels. The light levels are difficult to determine, but we're working at levels that you would get from normal daylight. Not much brighter, not much fainter. A sort of daylight, not direct sunlight levels, but daylight, the light you get from the sky. So that's where we're doing these experiments, or going to start these experiments. And that's another attempt to do what Kuru did some years ago and try and get an action spectrum of potentially different processes going on in this range of the spectrum where the body is relatively transparent.
Mm-hmm.
So I think this is the way to go, is to try and dig a little bit deeper into which wavelengths do what, and try and see the range of interactions that can occur between light and biomolecules.
Yeah, that's fascinating. I think there are a lot of people out there who just assume that we make energy from light because, you sort of feel better when you go out in the sun, you feel more energetic. So I think people already take it for granted that even if we don't know the mechanism, we certainly are generating some sort of energy from sunlight. Who knows?
Well, if we can take those people and convince them that blue light is not helping, that will be a move in the right direction.
Yes. Yeah, that's a tough one to get everyone to change all their lights in their houses and workplaces, but we'll get there. Yeah,
so I think we've kind of covered the balance between the blue light and the red light. There's another thing I should say, which Glen may have said to you. I have listened to Glen's interview with you, but he may have said to you, I can't remember. And that is, it seems that the blue light interactions with mitochondria work on a different timescale from the red light interactions. So we are dealing with it may be a different part of the same process, it may be different processes, but it takes longer to produce a reaction on biological cells with blue light than it does to get them to react to red light, which is kind of indicating that we're looking at either a different stage of an interaction or a different kind of interaction with these two colors. So although they seem to balance one another in effect, one mitigates the effects of the other, they're not working at exactly the same time scale. Which makes sense if you think what we've been talking about. The generation of reactive oxygens is likely to be different from the generation of antioxidants. I mean, there are different processes. So it's not nonsense. It does make some sense. But that's one of the observational elements that we have to fold into studying what's going on.
Yeah, I think there's just so much that is going to come out in the next decade about how biological systems interact with light.
Yeah.
Hopefully, there's a lot more people who are looking into the dangers of blue light and trying to get the big companies to change them, like you
It
are.
is it is happening. Yes, it is happening.
Yeah,
I can see evidence that people are beginning to take note now. I mean, I talk to a lighting engineer regularly, a lighting engineer in Silicon Valley, who's building multi-LED light sources to dial up different spectra. So he's using these, he's selling these to work in labs and work in medical environments where you
right.
can adjust the lighting in detail for different kinds of conditions, human conditions, therapies, and so on. And he's very helpful, and we talk a lot about these issues. And he's obviously in contact with the lighting industry, and he notices that people are beginning to take note of what's going on. And I think the change will happen. But it's like a huge oil tanker, isn't it? It takes a
Yeah,
lighting industry as a big industry, and it takes a lot of effort to change them even slightly in direction.
yeah.
Maybe,
I've often wondered also about, animal models, where they put rats um, in environments that are lit with completely artificial light, and then they look for health outcomes based on, what they're eating or
mm,
anything that they're feeding them, and they never mention the light. And
mm,
I often wonder, they do lots of sort of, and I've talked to vitamin D researchers about this, how they replicate the sun in models of skin cancer in rodents, and they use these solar simulators. And one of these researchers told me that the UV content of those solar simulators is 10%.
mm,
And I thought that's probably more than double what we're receiving
mm.
outside, and it probably doesn't have, well, it definitely doesn't have the far infrared that the sun is having either. So I often wonder how accurate some of these animal studies are that are completely lit by artificial light.
I
Um,
think I think that's a very grave problem. And I think we were guilty of this in the early days. We were using white LEDs as solar simulators, and we realized that we were completely wrong. This was a crazy thing to do.
mm,
Um, yeah, I think there needs to be a lot of tidying up in the way these experiments are done. We're not actually working on animals anymore. We're working on humans or ourselves, and
yeah.
it's not possible to get permission to work with blue light on animals anymore. It's very hard to, because it is realized that it's damaging. So we have to be very careful. I don't have any experience of all this business, but it's yeah, there's a lot of tidying up that has to be done. But I think you're right. It's going to be a very rich field of research
Mm.
over the next years. And I think, can I come back to this question of interdisciplinarity, which
I
I think
was
is so,
just about to say, let's hop
yeah.
back into why it's so important to share ideas in places where you may not be as well versed. I think sometimes we can find ourselves completely boxed in and not exposed to any outside, maybe creative ideas that may push a field forward. So, why have you seen it really important to work with people who are in different fields?
I do see it as important, and I'm trying to think of the best way I can express this. I think I've always been interested in I would call it now, I wouldn't at the time, the big picture story. And
Yeah.
I spend time some time talking to kids about the Big History Project. This project that Bill Gates and David Christian developed of teaching kids the history of the universe rather than the history of the 18th century. And I feel very strongly that we should bring up our kids with a much broader knowledge of what we know. Um, Now, I think early in school, they get this broad picture to some extent, but later on, there's a lot of specialization. And I think my kids did the European Baccalaureate when we were living in Munich, and they study 9 subjects to school leaving age rather than the 3 or so A levels that people study in Britain.
Hmm.
And they were told by British universities that they'd have to do remedial years in order to catch up. And that absolutely wasn't true at all. They didn't have to, but they had a much broader education in different subjects. And I think, especially with history, the history of the universe, we can paint a pretty good history of the universe. Okay, we don't know what dark energy and dark matter is, but we can paint a pretty good history of the universe from the Big Bang onwards, right through to the origins of life and so on. Now, not all of those steps are filled in, but there's a continuous process of, the Big Bang, the first stars, the origin of the chemical elements in stars, the production of organic molecules in space, the formation of planets, the origin of life, and so on, all the way through. There's a continuous wide-angle view. And I have to say that, the origin of the chemical elements is something that is not really taught, I don't think, in schools. Where do the chemical elements come from? We now know that the Big Bang only produced hydrogen and helium and a spot of lithium, but where did all the other elements come from? They came from stars and stellar processes. Now, the big paper that addressed that issue was 1955, a paper by Burbidge, Fowler, and Hoyle called B squared FH. Have you ever come across this paper?
I think you mentioned it to me when we spoke last.
Yeah, it's 107 pages in the Reviews of Modern Physics, and it does the whole caboodle. Or discusses the details of the origin of all these elements in stellar processes and stellar explosions, and it's a fantastic story. It's one of the great stories in science, and it's not widely understood by the general public, I don't think, how elements. I think we should we should certainly learn this at school. It's such an important, the origin of the periodic table. It's such an important thing. And I knew all those people. I knew Fred Hoyle. I knew both Burbidges, and I think I'd met Willie Fowler. I certainly didn't know him well, but so I'd cut all of those people, and it always stuck in my mind. This was a fantastic story, and but that contributes to this global picture, and it's the whole thing is a mixture of physics in the early days, then chemistry in the intermediate days, and now biology, and it's a real mixture, and now it's thoroughly mixed together. So I think one has to get rid of this idea of physics, biology, and chemistry now. It's all mixed together. In some sense, it's all physics, but I wouldn't argue that amongst chemists and biology. But in some sense, it is, because it's all down to quantum mechanical processes and so on. But so I think this broad view is essential, and I appreciate that I've been incredibly fortunate to have a long career in astrophysics, and use big telescopes around the world, use the space telescopes, and play a role in some of this research along the way. And it's given me a very broad angle view, of course, and I'm very conscious of that. So when I retired, I did make a conscious decision to break out a little bit and try and understand about the Earth, its evolution and biological evolution, and so on. And this was very much stimulated by the last couple of years when I was actually working. I supervised or co-supervised a young student from China who's from Beijing. And he came to us for doing 2 years of his PhD. He came to us with a set of observations of a lunar eclipse made with a 2-meter telescope in China. Very high quality data. And he used it to study the Earth's atmosphere from the light coming from the moon during lunar eclipse. And we had done some of this before with other people. But his data were extremely high quality, and he wrote a wonderful thesis in the end on many techniques of transit spectroscopy, looking at exoplanet atmospheres, and the details of the observational techniques that you needed to use in order to get the right kinds of data. Extremely innovative, one of the brightest people I've ever come across. And he went on to work in Heidelberg and Göttingen, and now on postdocs, and now he's gone back to China. But working with him for those last couple of years really stimulated me to think much more about the Earth and its atmosphere and geology. And So I kind of made a background decision to broaden my areas of interest. And I never thought I would really, I wouldn't actually do these subjects professionally, but I would learn more about them. And, working with Glen was a step in this process. And, we were working on this at the cutting edge in some sense. But I learned a number of things about the process of doing interdisciplinary research, at least at my age end of the spectrum. It's very hard to do this when you're young because you're fighting for survival. You have to write papers in your very narrow field in order to get your first postdoc and your first job and so on. So it's very hard to do when you're young. But if you have to retire early, I had to retire fairly early because the European Space Agency kicks people out at 60 or 62 if you're lucky. So I decided to spend my time looking at least looking at other aspects of science, and ended up with eyes and vision and mitochondria and light and so on. That was accidental, but and I realized that you feel afraid of going into a field where you know very little. If you walk into a lab where everyone's a visual neuroscientist, you think, oh my God, I know nothing about visual neuroscience. They're going to think I'm a complete idiot. But it's not like that. You have knowledge that they don't have, and they have knowledge that you don't have. And that knowledge is at the cutting edge. And so you don't have to go back to ground level and work your way up in some sense. It's your ignorance, it's my ignorance that was the key element in that process.
Yeah.
Because I thought about things in quite different ways. I said to Glen, do you realize that's the wavelength that chlorophyll absorbs? Because I knew it was, but he may have done if he thought about it. He hadn't thought about it. So, it's this kind of interaction which is so vital. And then subsequently, I was aware that when I was talking to this photodynamic therapist who'd been introduced to me by Glen in UCL, I was talking to him about things that actually been vital for what we were trying to do. And there'd never been this contact before because he was in another department, in a different building, a different part of London.
Yeah.
They can't communicate. They don't communicate. They're siloed in very, very narrow fields, and there's very little communication. And I have to say the universities are working against this at the moment because the university is absorbing so many students to get money. They're closing off all the common areas in the university buildings. So, they no longer have areas where people can meet for coffee and, different departments can talk to one another. They're shutting the staff canteen. So the lecturers and professors sit in their offices eating sandwiches at lunchtime. So they never communicate. Mm-hmm. So there's very little communication between different branches of science. And I think this has to change. And my single experience with working with Glen in UCL is that this is very exciting. This idea of putting people, an astrophysicist and a visual neuroscientist, you wouldn't dream of it, would you? But in fact, one of the first people I bumped into when I first went to his building in next to Moorfields, I looked at this guy. I know you. It's Chris Dainty. He was an astronomer from, I think it was Imperial College, while I was at the Royal Observatory in the 70s, developing adaptive optics for astronomy. So, sharpening up the images from ground-based telescopes. And now he was working in a department of ophthalmology, doing adaptive optics for high-resolution imaging of the retina, a living retina. So he could counter the motions and the noise in the imaging of retina. And so there we were, astronomers together at the RGO in the 70s, sitting in the Department of Ophthalmology, working with light. So it does happen. It can happen. But we have to make it happen more. And we have to also dissolve the boundaries between the sciences, I think, and try and breed a broader culture of expertise in scientists. And I think at my age, it's somewhat easier to do because, there are a lot of retired people around who have a great deal of expertise and knowledge in different areas who could come together and tackle some of these problems. If you really wanted to solve if it were wartime and you really wanted to solve a problem, Churchill during the war used to grab people from all over the place, stuff them in a room and say, fix the problem.
Yeah.
And, they'd do it. And they were a great mixture of different people. People would solve crossword puzzles or, build radar sets or whatever. You have to bring them together to solve these fundamental problems. And so I think this is a new way that science could and should develop. And I feel I have to say something about my experience of interdisciplinarity. My experience, my first author paper in biology, I wouldn't say it hit the headlines, but it certainly had a significant impact, much more than I would have expected.
Yeah.
Why is that? It's because, what I said was trivial for physicists, but unknown to biologists. And, it wasn't my great brilliance at all. It was just bringing these two subjects into contact, which makes the difference. And, it's worth thinking about, I think.
Oh, absolutely. I couldn't agree more. And I'm sure there are countless stories out there about how innocuous comments from people who aren't entrenched in the field completely change the way that you might see and think about a topic that you've been working even years on. I know we spoke a little bit last time about water and Gerald Pollack, who spent years thinking about where the energy came from that created this particular type of water. And it was one of his students who was being silly and not doing what he was supposed to do and pointed a lamp at the water during an experiment and realized that the infrared light from the lamp was what caused the water to make that change. So I'm sure there's plenty of stories where it's things like exactly the story that you described, where something that was trivial to you really clicked into place, thoughts in the minds of the people who've been thinking about it for a very long time. It
does happen. It does happen. It does happen. So I'm not saying it doesn't happen. It happened to Raman. When Raman discovered Raman spectroscopy, when his student was shining a sunlight through a flask of something, and he whipped out his pocket spectroscope. Raman whipped out his pocket spectroscope and looked at the flask and saw all these lines, and that was it.
Mm-hmm.
And I love that story. And it is an example that's. It's not quite the same. It's not quite so interdisciplinary, but it's keeping your eyes open for unexpected things. And and I got maybe we could finish off. I tell you another story about amazing coincidences, which actually blew my mind. And I am an astronomer, although I'm not very actively an astronomer at the moment. I do keep my eye on the literature. And I was bowled over to see the first images coming from the Webb telescope a couple of months ago now, and there were a batch of them produced in the first week and discussed by the President of the United States and so on. But about a week later, they produced an image of a galaxy called the Cartwheel. Which, of course, I recognized because I'd named it the Cartwheel with a collaborator of mine in Australia in 1976. And so this amused me, and I wondered why they'd chosen the object. It didn't seem to be an obvious object to choose for one of their early release observations. But I immediately afterwards went on holiday. And while I was on holiday, I received an email from somebody I knew from those days who was a mathematician at MIT. He's still a professor of mathematics at MIT, applied mathematics at MIT, called Al Tumri, who is a very well-known astronomer who was really the originator of ideas about the dynamics of galaxy interactions. So he was the person who started modeling the interactions between galaxies and produces these wonderful interacting galaxy shapes and tidal tails and so on that we will now see with these astronomical pictures. And the story there was that I was working with somebody who worked for the United Kingdom Schmidt Telescope on Siding Spring in Australia. And I was working at the Anglo-Australian Telescope. And, you know, we were on the same mountain, and we knew everyone there. And the Schmidt, they just rediscovered this galaxy, which was in the form of a ring, which is extremely unusual. And they realized that it was a rediscovery because it had originally been discovered by Zwicky, the very famous Swiss astronomer, in the 1940s. In fact, he mentioned it in a paper in 1941, which is the very first reference about the discovery of this galaxy. And he mused that while he didn't understand how this was produced, it was probably one of the most complex structures that he knew about that needed to be explained by some theory of dynamics or whatever. It subsequently appeared in a catalogue by Vorontsov-Belyaminov, who was a famous Russian astronomer at the time, and so it was catalogued, but it was essentially rediscovered when the Schmidt people were looking at their plates every morning, as they always do. And so I got together with Tim Harden, and we had some time. I had some time on the Anglo-Australian Telescope. We had actually a few very good nights, very clear nights. So we observed this, and we had, at the time, we had the world's leading electronic detector, spectroscopic detector, because our director was Joe Wampler, who developed the Wampler-Robinson scanner, which was one of the very first digital electronic spectroscopic detectors. So we had the 4-meter telescope and a state-of-the-art detector operating. So we did spectroscopy of the various components in the ring and the neighboring galaxies of this cartwheel ring galaxy. And while we were writing up the paper, I got a preprint of a theoretical paper by Roger Linz and Al Altumre about interacting galaxies, which actually had a simulation of what would happen if one galaxy interacted with another one by falling directly through the center and out the other side. So it's a very symmetric collision where you come along on the axis of a spiral galaxy, you drop right through the center and emerge on the other side. And he had theoretical predictions of what it would look like and so on. And I looked at this and I thought, golly, this is exactly what we're seeing. So we measured the rotation speed of the ring. We measured the radial motion of the ring, which was either expansion or contraction. So we had a rotating ring, which was either expanding or contracting. We surmised, because of the direction of the spokes, which you can see in the cartwheel image, that it was an expanding ring. And we did quite a lot of basic calculations about what the timescale was, how long it was between the actual collision itself when the intruding galaxy was in the center and reached where it might be now, and compared that with the timescale of the ring expansion, and they agreed to about 300 million years. So it looked as if one of the galaxies that nearby was the intruder that had gone through the center. And we justified that, and Al Altumre agreed that it was the likely intruder, and we essentially solved the problem at the time. There was one glaring discrepancy, and that was that his models suggested that the mass of the intruder had to be at least a third or a half of the mass of the original cartwheel galaxy before it was stimulated to produce the ring. And it was not clear that this intruding galaxy was massive enough. In fact, it was measured by one of my colleagues at the time later, and the subsequent director of the observatory. They measured the velocity dispersion of the stars in the center of this proposed intruding galaxy, and it seemed that it wasn't massive enough to cause the interaction. So this was a problem that we didn't solve at the time, and it remains a problem. And when the galaxy was the image was announced by NASA, there was quite a long press release that came with it. It didn't mention us, of course, but it did say that there was a more distant galaxy about 3 times further away, which they thought was the intruder, which had been proposed before, I remember, but not with any strong evidence. And so when the image was announced, they said the intruding galaxy is not in the image. Excuse me. It was not in the image. It was outside the field of view of the image. And I thought, well, it is in the image. And Allah wrote me an email and said, well, it is in the image. So Allah and I have been having lots and lots of email exchanges over the last few months, and he's rebuilt his models, and we're remodeling it now. But that argument's going on. But that's an interesting story in itself. But the real story is that Tim Harden, my collaborator at the time, when we both left Australia subsequently. I went off to do my career in Geneva and Munich and so on. He went off to Edinburgh and worked on the technology of infrared instruments and telescopes. And unbeknown to me, he was working on the ideas of passively cooling space infrared telescopes, because he realized, to get to these very low temperatures you needed for infrared telescopes, you had to cool them to very low temperatures. And a number of observatories have been launched into space with limited amounts of coolant. So the telescope Herschel, IRAS, and so on. These would only last for a limited time because you had to cool everything to low temperatures.
Mm-hmm.
And so he had this idea of using a sun shield and passive cooling of the telescope to reach, relatively low temperatures. Not the very coldest temperatures you needed for longer wavelength infrared, but certainly for, near to mid-infrared, you could cool the telescope enough passively. So the story, as I hear it, is that he tried this idea out on the European Space Agency, and they didn't have any applications for it at the time, so they weren't particularly interested. But he took it to NASA, and this must have been in the early 2000s. I'm surmising. I don't know the story in detail, but he took it to NASA, and they said, "Hey, this is a good idea. Maybe we could use it. We could try it out on Spitzer." And it must be earlier then. They tried it out on Spitzer, and it obviously worked well enough for them to use this for JWST. So the whole design of JWST, with this huge tennis court-sized sun shield, mm-hmm. and the passive cooling of the telescope to 40 kelvins, came from Tim Harden, who worked on the cartwheel with me. And my wife found this. She Googled, we were talking about this at the dinner table, so she Googled Tim Harden to find out what had happened to him. I knew that he died in 2009. He had a heart attack and he died in 2009. In 2010, he was awarded a very major NASA engineering award for the design of passively cooled telescopes that they used on the Webb telescope. And it was a year after his death, and it was presented by John Mather, senior project scientist of JWST and the Nobel laureate. Mm-hmm. So this is written up. It's actually on the web. It's written up. This award was given to Tim Harden. So there's this, I think, extraordinary historical loop between the cartwheel and the use of the James Webb to image the cartwheel as one of its early release observations. And I don't think there was an intended connection there. It's just an amazing coincidence. It may be that somebody in NASA said, "Hey," but I very much doubt that, because I didn't think they knew about our paper at all. But it blew my mind at the time, and I thought this is just an amazing coincidence. But so now we're working quite hard on this now. So the person who measured the velocity dispersion of the intruder and said the galaxy was not massive enough, we're in contact with him. In fact, I got an email from him this morning. So we're going to look at this whole problem again and try and understand it. But I'm pretty sure that we have chosen the right object, and we have to understand why it produced such an effect. But that's fun. So it's an interdisciplinary universe that I live in.
Yeah, that's an incredible story.
Yeah.
I wouldn't have had any idea that I'm astonished that that's not on purpose. If it's not on purpose, it's an amazing coincidence that they chose that.
I'm pretty sure it's a coincidence. But I will try and dig out more about the story. But because I know where the proposal, I now know who proposed the cartwheel as a potential target for JWST, and it's somebody I think wouldn't know about that coincidence.
Right.
So I think it is a coincidence, but it is an amazing story. And to think that somebody beavering away in an office in Edinburgh can think of something as with as high impact as the design of the Webb telescope is pretty astonishing. So my contribution of the wavelength calibration standard pales into absolute insignificance compared with his contribution, but it's a fascinating story. But it shows you how many links there are between these different things, and how it sometimes crystallizes out of history long after the event.
Absolutely. Yeah. Wow. We've covered a lot of ground, haven't we? We've
probably gone on for long enough, haven't we? But
was there anything else you wanted to go through before we wrap this up?
I think, well, I told you in the main areas. I think they were the broad background, the wide-angle view, the interdisciplinarity, the fact that we need to encourage that interdisciplinarity. I was puzzled why all of this work on light and biology happened in departments of ophthalmology, which I now understand, but I didn't understand in the beginning, because you can get light into the eye. That's a very important issue, I think.
Mm-hmm.
Otherwise, I think, yes, you mentioned this problem about water, and I do think that's very interesting. I did look at it, as I said to you, I did look at that at the time. I think it's possibly relevant, possibly highly relevant, but I don't know the answer, and I don't really have any means of investigating it, I think. But I do think there are processes going. I think biology is complicated. I know biology is much more complicated than physics. And when you try and isolate a particular phenomenon, you find that many other phenomena are contributing to the same process. Yeah. And I think that's a hard lesson we have to learn about biology. And it may well be that these perhaps slightly unexpected processes, like the influence of slight heating or water or light in some way on the structure of water, could well be a significant contributor. And water is a wonderful story. I actually knew one of the stellar dynamicists who I grew up with was somebody called Donald Lyndon Bell, who was a well-known theorist at Cambridge, and he was around when I was a student. In fact, he examined me both as my interview board for my PhD and also for my progress through my PhD.
Wow!
In the chapel at Herstmonceux, I remember I had an examination, and these were oral examinations, which were quite terrifying. And I remember one question he asked me. He said he was pacing up and down the back of the chapel at Herstmonceux, and he said, "Bob," he said, "I've got this electron, and I hit it very hard with a cricket bat. What spectrum does it emit?" Anyway, So his wife was working on water and water structure. In fact, she was the editor of one of the major tomes on the properties of water. So this is another coincidence that goes back to. I knew his wife slightly. I knew him quite well, but I knew his wife slightly. But she was one of the workers on water. I'm conscious of the fact that water structure is very complex and absolutely fascinating, and it certainly, I think, there's without doubt, it contributes to biological processes in many ways. And this may be one of the ways. I mean, the reason I was somewhat hesitant about it is I don't think I would have chosen that wavelength if I wanted to influence water, because there isn't a strong water band at 670 nanometers,
Right.
and all the stronger water bands are longer wavelengths. So it's not entirely obvious to me that that would be the process one would choose. But and when you get down to very thin layers of water, the absorption coefficient of water at those infrared wavelengths is very, very low.
Mm.
So I don't know, but I can't really. I don't think I can say much more about it. But I hear it. I hear what you say, and I think it's potentially very relevant or potentially relevant. But I don't understand how it could work under this circumstance.
Yeah, I'm not sure anyone's got a firm grasp on it at the moment. Water seems to be very ephemeral and
Hmm.
hard to sort of get your hands around. And anytime you think you're onto something, something pops up that makes you think otherwise. But yeah, I just missed the International Water Conference. It was in Germany a couple of days ago. I wanted to go this year, but unfortunately, I was unable. I'll try and make it next year because there's a lot of brilliant people there talking about cutting edge stuff. Lots of things related to water being conscious, water being able to sort of understand its environment and wacky stuff like that.
Yeah, I've done a lot of experiments on water myself. I managed to observe the Raman spectrum of water with my spectrometer. I don't have a Raman setup, but I managed to fudge it so I could observe the entire spectrum of water right the way through from the deep ultraviolet right through to about 6 microns and the fundamental bending mode of water, just with the
Wow.
spectrometer on my desk and a laser pointer that I had. So I love observing water, and it's absolutely fascinating. But so I'm interested in the structure as well. But
Time will tell.
time will tell.
Yeah.
Anyway,
All
well, it's
right.
been a delight to talk to you. I don't often get the opportunity to expose my life in this way, but
No, look, in the interest of
it's quite fun to do.
interdisciplinarity,
And
I think I had an awesome time talking to you. I was nervous, trying to psych myself up to make sure I didn't say anything stupid, but I think we got through that all right. Um, yeah, so I think it's great what you're doing at the moment with Glenn's group. I really hope you continue to do this work because I think it's actually making quite a big difference, and a lot of people are becoming interested in particularly the health effects of both red and blue light. And even if we can get one person to sort of change their habits and get things a little bit more dialed in as far as avoiding poor spectrums, that would be fantastic. So
well, I would perhaps complement that and end the message by saying to people, if you want to buy LEDs to use at night in your house, don't buy the high temperature ones, buy the low temperature ones and have the warm yellow lighting and not the bright white lighting. I think if we could get that one message over, that would do a lot of good. So
yeah, absolutely. I couldn't agree more. And I think the work that you're doing is only going to continue contributing to people understanding that this is something that's very important and can't be ignored anymore. So yeah, let's hope we can reach as many people as possible. And yeah, keep doing what you're doing because it's very meaningful. And yeah, thank you very much.
okay, thanks, Cameron.
All right, Bob.
Oh yeah, very, very pleased to talk to you. Okay,
Yeah, it
all
was
the best.
awesome. I'll talk to you soon.
Okay,
Bye
cheers.
bye. Thank you so much for listening to this episode. I sincerely hope you enjoyed it. If you'd like to keep up with Bob's work, I've put links to his profiles in the episode notes. He has an excellent Flickr page that I would really encourage you to check out. If you'd like to support this podcast, you can subscribe on Spotify and YouTube and leave up to a 5-star review on Apple Podcasts. This is a simple, no-cost way to support my work and help me reach more listeners. Please feel free to leave any comments on my YouTube channel as I do try and read through as many as I can. I've also put links to all of my social media platforms in the episode notes if you'd like to keep up to date with the podcast, get information about health, or if you'd just like to reach out to me in general. Thanks again for listening, everyone. Take care.